Hello, everyone, and welcome back for more human physiology. So, at this point, we have already talked about passive transport as much as we're going to talk about it. So, now it's time for us to move on to the other kind of membrane transport, which is called active transport. So, the way that we defined passive transport is that it involves all movement of molecules across the membrane when you're going from high to low concentration. And as we've already established, that means that you do not require energy, regardless of whether you're talking about simple diffusion or facilitated diffusion. Because those molecules are going downhill, because they're going down their concentration gradient, there is no energy requirement. Well, active transport, the story is different. Active transport does require energy. In most cases, this is going to be because we are trying to push the molecule against its concentration gradient. We're trying to go uphill. And as we've talked about before, if you try to go uphill, then you're going to have to give it a little bit of a push. You're going to have to expend some energy. So there are two major types of active transport. The first that we're going to discuss here is basically going to involve what I just said, pumping a solute from an area of low concentration to an area of high concentration. Again, you want to think of us going uphill. This is still going to utilize a carrier or a channel like we are already aware of for facilitated diffusion, the main difference being that we're going uphill from low to high concentration and we're going to need some form of energy. Second, this is going to be part of your on-your-own work, importing or exporting something that is very, very, very large, so something as big as a protein. So as it turns out, using facilitated diffusion or even using channels or carriers at all, really that's only going to be useful for small molecules. If you've got something really big, you're going to have to use one of your modes of bulk transport, exocytosis, endocytosis, or something similar. So we just call them all bulk transport. They do require energy, but we're not really going to get into it here. Okay, so when it comes to active transport, most commonly you are going to be talking about moving ions across the plasma membrane. At this point, we are already familiar with sodium and potassium. In particular, we are familiar with the idea that the concentrations of sodium and potassium are very different when you compare the ICF to the ECF. So you'll remember that the intracellular fluid has a lot of potassium but not much sodium. The extracellular fluid is just the opposite. The extracellular fluid has a lot of sodium but not so much potassium. So have you wondered why is it that way? Why do we keep things that way? Well, there is actually a very special kind of active transporter that is always functioning in every cell in your body to make sure that these fluids stay the way that they're supposed to be. So this transporter is called the sodium-potassium ATPase pump, or you can just call it the sodium-potassium pump. So this protein, this transporter, will help to maintain these concentration gradients across the membrane for both sodium and potassium by using ATP molecules. At this point, we already understand pretty well that ATP is a very useful and common form of energy for cells. So this is just a case of trying to move both sodium and potassium at the same time against their own respective concentration gradients. And the cost that we have to pay to do that is a molecule of ATP. If you look at the diagram here, you can actually see this transporter, which is represented by this kind of oval here. It doesn't actually look like that. We're just trying to keep things simple here. So basically, any sodium that is found inside the cell, and keep in mind there shouldn't be very much sodium, but there should be a little bit of sodium. This transporter is going to take what little sodium is in the cell and pump it out into the extracellular fluid where the concentration of sodium is higher. Similarly, with potassium, we take what little potassium we can find in the extracellular fluid and we pump it inside. Now, how this actually occurs is a little bit more complicated. It's a stepwise process in which we get the sodium going first and then the potassium. You don't need to worry about that. All you need to worry about is the end result. essentially 3 sodiums leave the cell at the same time that 2 potassiums come in. And to accomplish that, we are using a molecule of ATP to pay the cost for both of those ions moving against their concentration gradients. So what we have just described here is a type of active transport called primary active transport. This is reserved for active transport processes that are getting their energy specifically from ATP hydrolysis. So you're probably wondering, well, where else could we get the energy? Well, that's a very good question. On the next slide, we are going to look at secondary active transport, which is the other kind of active transport. So this means that we are not going to get our energy directly from ATP, but from indirectly a different source. Now, the best example of secondary active transport in action comes from how we absorb nutrients through the intestinal tract. Specifically, we are going to look at glucose here. Now, I will tell you, this is a extremely important process. I guarantee you're going to see questions about this on the exam, so definitely I would pay a lot of attention here. The reason why this is such a useful process for us to look at is because this whole process of absorbing glucose from the food that we eat into the bloodstream actually exemplifies 3 of the 4 kinds of transport that we have actually already looked at. Okay, so before we get into specifically what's going on here, let me set the stage for you. When you eat something, so when you eat a meal, all that food is going to get digested in your stomach, meaning that enzymes are going to start breaking it down into individual subunits. Carbohydrates get broken down into monosaccharides like glucose. Proteins start getting broken down into individual amino acids. Lipids get broken down into fatty acids and things like that. So by the time that digested material hits the intestines, the intestinal walls are fairly thin, and on the other side of the intestines is going to be the blood supply, and that is what we want to accomplish here. We want to absorb these nutrients from the food that we have digested, which are currently in what we call the lumen of the intestine, basically the inside hollow part where all the food is and all the digested materials. We want to absorb those nutrients into the bloodstream, which is on the opposite side of these epithelial cells that actually make up the intestinal wall. What this means is that in order for these nutrients to make it from the lumen of the intestine and into the bloodstream, there are 2 different membranes that they are going to have to cross. They first have to cross this top membrane up here, which has these little microvilli structures here that go up and down and up and down. This is called the apical membrane of the epithelial cell, and you will learn more about this in Chapter 4. Then, from there, once we cross the apical membrane, that nutrient molecule is going to be inside the intracellular fluid of that cell. Well, that's not really where we want the nutrient. We want it down here in the extracellular fluid, which is going to include the blood plasma. So that means once the glucose in this case is inside the cell, it needs to cross down through this bottom membrane, which is called the basolateral membrane. Again, you'll learn about that in Chapter 4. So if we're going to absorb glucose from up here in the lumen to down here in the bloodstream, that glucose needs to cross the membrane up here at the apical membrane and then down here at the basolateral membrane. So okay, we know what we need to accomplish. So the first thing for us to do is check our concentration gradients to let us know what is going to be necessary. The first thing to know about glucose, obviously, is that it is very polar, so it cannot use simple diffusion. We're going to have to use a carrier or a channel to let it through a membrane, regardless of whether we are using passive transport or active transport. So now, if we check our concentration gradient, even though we have eaten food and we've got this glucose in the lumen of the intestine, there is actually more glucose inside the intracellular fluid. So we have got an unfavorable concentration gradient here for glucose. If we want glucose to cross the apical membrane, we are going to have to push it uphill. We're going to have to go against the concentration gradient. That means we are going to have to use active transport. Now, we've already looked at primary active transport, and we could utilize a molecule of ATP in order to get glucose across, but that is not actually what we're going to do. We are instead going to use secondary active transport. Well, what is secondary active transport? So, secondary active transport will involve one molecule moving through a carrier at the same time that another molecule moves through the same carrier. What we're specifically looking at is that one of those molecules, glucose in this case, is going against its concentration gradient. It's going uphill. The other molecule or ion that is going through the channel at the same time actually has a favorable concentration gradient. It's going downhill. So if you look at this channel here, which is called the SGLT carrier, it is letting glucose through at the same time that it is letting sodium through. You'll recall that there is more sodium outside the cell than there is inside the cell, so this works out perfectly for sodium to enter the cell. It's going down its concentration gradient at the same time that glucose is going against its concentration gradient. Essentially what is happening here is that sodium is paying the cost for glucose. When an ion or a molecule moves downhill, it actually releases a little bit of energy. Usually we're not able to harness that energy, but in this case we actually can. Sodium moving downhill provides the momentum needed for glucose to move uphill. That is secondary active transport. It involves a molecule moving up its concentration gradient, and that cost is paid by a different solute going down its concentration gradient. Either way, we are still using energy to pay a cost. The difference here is that we are not specifically using ATP energy directly, although I will have something to say about that a little bit later. Ultimately, the energy does come from ATP, but it's a lot more indirect, so I'll have more to say about that here in a minute. Okay, by using secondary active transport, by using this SGLT carrier, we let glucose inside the cell. So now we've got a bunch of glucose inside the cell. That's great. We've crossed the apical membrane, but we know now that we also need to cross the basolateral membrane. Okay, let's check our concentration gradients again. We have a high concentration of glucose inside the cell and a low concentration of glucose down here in the ECF. Well, that's great. We actually have a favorable concentration gradient. So all we need to do here is set up a transporter to let glucose through, and that's what we're going to do. So glucose is going to move through this carrier, and that is facilitated diffusion. Nice and easy. We don't have to use energy because glucose is moving downhill in this case. We deliver glucose down here into the bloodstream, and ultimately that is what we were trying to accomplish. So that's it, right? That's all we need to talk about here, right? We got glucose across the apical membrane. We got it across the basolateral membrane. Glucose is in the blood now. Game over, end of story. That's all we need to do, right? Well, no. That is all we need to talk about if all you're ever planning on doing is just a little bit of glucose absorption. Eventually, if we do this enough, we are going to lose our ability to absorb glucose, unless we talk about something else. Now, let me introduce you to what I'm talking about here. Let's go back to the SGLT carrier up here. Every time we let a molecule of glucose through, we also move a sodium ion down its concentration gradient. We know that we have this concentration gradient for sodium, and we don't want to take that for granted. Again, think about it. Every time glucose moves through, we lose an ion of sodium from the extracellular fluid and we place it in the intracellular fluid. Every time we do that, the concentration of sodium in the extracellular fluid goes down and the concentration of sodium in the intracellular fluid goes up. So essentially, if we transport enough glucose this way, we are eventually going to reduce the amount of sodium in the ECF and increase the amount of sodium in the ICF so much that we are going to establish equilibrium for sodium. Eventually, we'll get to the point where there's an equal concentration of sodium inside and outside the cell. And if we do that, guess what we're not going to be able to do anymore? We are not going to be able to transport any more glucose. So we can only bring glucose across the apical membrane if we have a concentration gradient for sodium. So the way we solve this dilemma is by using the sodium-potassium pump that we introduced on the previous slide. On the basolateral membrane, you have this sodium-potassium pump, and what it does is every time we bring sodium in by using the SGLT carrier, the sodium-potassium pump says, nope, you're not supposed to be there, and it kicks the sodium back out into the extracellular fluid where it belongs. And we also bring potassium inside the cell. By doing this, we keep the concentration of sodium low inside the cell. Sodium can naturally get replaced in the up here in the lumen just from the salty foods that you eat, but we have to make sure that we're reducing the amount of sodium in the intracellular fluid. And having the sodium-potassium pump accomplishes just that. We make sure that we maintain a strong concentration gradient for both sodium and potassium, because these sodium and potassium concentration gradients are used for secondary active transport. Secondary active transport is not possible if you do not have these strong concentration gradients. And the sodium-potassium pump is responsible for maintaining the concentration gradients for both sodium and potassium. As long as we've got those gradients, we've got a store of potential energy that we can tap into if we ever need to move something like glucose against its concentration gradient. So the takeaway here is that you're not always going to have a favorable concentration gradient. If you need to pump something against the gradient, you're going to have to use primary active transport. And whether you're using whether you use primary active transport or secondary active transport, that's just going to depend. Okay, so one last thing that we can do here is look at some of those transporters that we were talking about and introduce a little bit of extra terminology here. We can call transporters and carriers depending on how many molecules or how many solutes they're letting through and in which direction. So the first type of carrier is called a uniport carrier. Uni, of course, is a prefix meaning one, so a uniporter is going to be a carrier that only lets through one solute at a time. Doesn't really matter which direction it's going. It could go from ECF to ICF or ICF to ECF. If you're only letting through one substrate or one solute at a time, you've got a uniporter. The best example of this on the previous slide would be the facilitated transporter for glucose in the basolateral membrane. That is uniport that we are seeing right there. The next two will involve two solutes moving simultaneously through the same carrier, but we call them different things depending on in which direction both molecules or both solutes are going. If both solutes are moving through the carrier at the same time and in the same direction, we call that a symport carrier. Sym is a prefix that refers to two things being on the same side of something. Sodium and glucose here are on the same side. So the SGLT carrier that we saw before, that brings sodium down its concentration gradient and glucose up its concentration gradient, is a perfect example of a symporter. The sodium-potassium pump that we saw before, which pumps sodium and potassium in opposite directions, is an example of an antiporter. Anti is a prefix that means on opposite sides, so it therefore makes sense that these two solutes would start on opposite sides and then move in two different directions. All right, and then the last thing that we want to look at here is just a quick little case study. Pathophysiology is very important for us to learn and understand and appreciate as we go throughout the semester. There's no point in understanding how the human body works normally if it never breaks down and we never get disease. And this is a particularly nasty disease. This is called cystic fibrosis. This is a genetic disease, so you'll recall that before you'll learn later on in this chapter about genetic diseases and mutations. Specifically, cystic fibrosis is going to result from a mutation in a gene called CFTR. So a gene is just a set of DNA instructions on how to build one protein. And the protein that we are building here is a chloride transporter. This transporter is going to pump chloride ions into the alveolar fluid of the lungs. This is a fluid that is secreted into the interior part of the lungs, the lumen of the lungs. It helps to lubricate things and it helps to make the breathing process nice and easy. So normally, there are several different membrane transport processes occurring here. So when we let chloride transporters into the lumen of the alveoli, sodium ions will follow through a separate channel because of an electrical attraction. The chloride ions are negative and the sodium ions are positive. Well, if we do both of these things, we move two different solutes across the membrane, there's going to be a big osmolarity change between the two sides. So osmosis is going to occur to correct the difference, so we end up secreting some water into that space inside the alveoli, and that helps to provide some nice lubrication, and like I said, it helps to make the breathing process nice and easy. Well, the problem with the mutation that we see in this CFTR chloride channel is that we don't make the protein correctly, and the cell will actually destroy the channel before it ever has a chance to do its job. So the chloride ions don't get transported, the sodium ions don't get transported because there's no chloride ions to get attracted to, the osmosis never occurs, so we don't do a good job of putting water into that secretion in the alveoli, so it becomes very thick and very viscous. It's like having a really thick paste inside the lungs. And I'm sure you're aware, if you've heard of cystic fibrosis, that in addition to producing digestive issues, it also makes breathing very, very, very difficult. So here you can actually see an example of a particular pathophysiology that is especially defined by a problem with membrane transport. We're not talking about membrane transport just for fun. Membrane transport is fundamentally exquisitely important for many, many physiological processes. All right, that is going to do it for this video. Thanks as always for your attention, and I will see you next time. We will start talking a little bit about cell organelles, and in particular, we are going to focus on mitochondria, ATP production, and cellular respiration. Thanks again, and I will see you next time.